So what we cover today will be on the exam on Wednesday, so we pay attention. So what we're doing today is we're going to finish the story about macrophages as professional antigen-presenting cells. We started that on Friday, and we went almost all the way through it. But what I want to do is I want to leave you with the idea that these macrophages will express on their surface a kind of protein, a special protein called MHC2. MHC2 stands for Major Histocompatibility Class II proteins. MHC2 proteins are only found on 3 cell types. They're found on macrophages, dendritic cells, and naive B lymphocytes. We're going to talk about the story of macrophages because the macrophages that we introduced last time were participants in the innate immune response of inflammation. But these macrophages now are going to form a bridge between the innate immune response and the adaptive immune response. And we'll see that these macrophages, because they contain MHC2 proteins, are said to be professional antigen-presenting cells, and there are only 3 cell types that do that. Macrophages, a cousin called the dendritic cells. And if you take a course in immunology, you'll find that these dendritic cells play an even more important role as antigen-presenting cells than macrophages. But the story fits nicely with macrophages because we can tie in the idea of the monocyte arriving at the site of inflammation, proliferating, then maturing into a macrophage, gobbling up foreign proteins, and then becoming a professional antigen-presenting cell that goes out into the germinal regions to present those antigens. But there's another character we will talk about today, and that's the naive B lymphocyte. The naive B lymphocyte is also considered to be a professional antigen-presenting cell. And what ties all 3 of these together is they contain MHC2 proteins. We're going to emphasize the idea of the interaction of the naive B lymphocyte with another cell called a T helper cell. That's part of the cellular branch. When the naive B lymphocyte interacts with the T helper cell, that will tell this naive B lymphocyte to divide and make millions of copies of itself. And each of those copies will have an identical antibody, that antibody that was generated from the genome of the naive B lymphocyte. And we'll talk about that. When we talk about cells of adaptive immunity, we're going to talk about the humoral branch. Whenever I say humoral branch, immediately think plasma B cells, or at least a B lymphocyte. The plasma B cells are going to be mature cells that have been derived from naive B lymphocytes. When you think about the humoral branch, we're going to emphasize the idea that these B lymphocytes, once they're mature, once they're plasma B lymphocytes, these mature plasma B lymphocytes are secreting a glycoprotein called an antibody. So hold on to the idea that the antibody is secreted by the B lymphocyte. The antibody then binds to a foreign protein. That foreign protein is called an antigen. So the antigen is foreign, the antibody is self. When the antibody binds to the foreign protein, the antigen, it forms a 3-dimensional structure called an antigen-antibody complex. And that's basically, and this binding of the antibody to the antigen will help inactivate that antigen. And we'll talk a little bit about that. But the humoral branch does not work alone. It has to work in concert with the cellular branch. And we'll see a little bit about how this is going to work. When you think humoral branch, so we're still on that panel, when you think humoral branch, think secreted antibodies. When you're talking about the cellular branch, however, the first protein that needs to come to mind is something known as the T cell receptor. The T cell receptor is a transmembrane. It's always a transmembrane protein. And the T cell receptor can only bind to foreign proteins called an antigen. It can only bind to antigen fragments that are on other cells. Now, there's a story here in that if we're talking about CD4 cells, if we're talking about T helper cells, so whenever I say T helper, think CD4. Whenever I say T helper, think it will only bind to professional antigen-presenting cells. It will only bind to professional antigen-presenting cells because it has to recognize MHC2. And we'll see that CD4 recognizes MHC2. Their cousins, which have come through the thymus, are the CD8 cells. CD8 cells are also known as T killer cells. But these CD8 cells, like their cousins, the CD4 cells, these guys have T cell receptors in the same way. So whenever you think about the cellular branch, think T cell receptor. The T cell receptor is going to recognize foreign proteins. They're going to recognize foreign antigens. But unlike the B lymphocytes, which secrete antibodies out into the world at large that can bind to whole protein, these are restricted to binding to antigen fragments. Not only are they restricted to binding to antigen fragments, but those antigen fragments have to be presented to these cells in a particular way. For the CD4 cells to recognize foreign protein, that foreign protein needs to be presented on MHC2 proteins. For CD8 cells to recognize foreign proteins, that foreign protein needs to be presented on something called MHC1. Now again, hold on to the idea that MHC2 is only on a very small subcategory of proteins: dendritic cells, macrophages, and naive B lymphocytes. But when we're talking about MHC1, every one of our cells, if it has a nucleus, now erythrocytes don't have nuclei, so they don't count, but everybody else is expressing MHC1. And what you're doing day in and day out is you're taking proteins from your cytoplasm and putting them on these MHC1 proteins, trafficking those MHC1 proteins to the surface, displaying them on the surface, and asking for T killer cells to come along and inspect them. As long as those proteins are self and things are working right, the T killer cells won't recognize them. But if it happens to be a virally infected cell and the viral proteins being displayed on the MHC1 proteins, then when the T cell receptor comes along, if it recognizes that antigen fragment, that fragment of the viral protein, it will kill that virus-infected cell. And if there's time, we'll go through that. When you think about antibodies in the humoral branch, again, these are glycoproteins that are secreted. We're going to talk about the antibody structure. We're going to talk about how the antibody works. We're going to talk about how the antibody binds to antigen. And then we're going to talk about how these B lymphocytes in the humoral branch, how do they originate? Where do they come from, and how are they selected? And that's going to be something called clonal selection. And we'll see, we're going to start out with a naive B lymphocyte. This baby naive B lymphocyte born in the red bone marrow goes out into the germinal regions. It takes residency in the germinal region. And then for it to function, for it to be useful, it has to interact with one of these CD4 cells. And we'll talk about how that happens. If this naive B lymphocyte binds to a foreign protein, it then becomes a professional antigen-presenting cell and talks to T helper cells. And the T helper cells say, yeah, I recognize that protein as foreign. Here's a bunch of growth factors, so go forth and multiply and make many copies of yourself. And each of those cells will have this, each of those daughter cells will have the same antibody as we started out with on the naive B lymphocyte. But let's start here with the idea of the macrophage as a professional antigen presenting cell. So here's the macrophage. Has this nucleus. I won't go through the whole process, but you may remember from last time we talked about transmembrane proteins binding to a bacterium. Then we pulled that bacterium into the cell. It went into a vesicle. The vesicle went to the lysosome. From the lysosome, we went to an endosome. In the lysosome, this bacteria was chopped up into its component parts. Basically, it was all these proteins, and all these proteolytic enzymes in the lysosome broke it down into tiny pieces. And then, out here, in the lysosome, there was this protein, special protein called MHC2, major histocompatibility. Class 2 protein. And its function out here in the endosome was to bind to one of these fragments of the original bacterium. So this is a protein fragment. It's in fact an antigen fragment. Typically, these antigen fragments are about 16 to 20 amino acids long. So they're short sections of a gigantic protein. But this endosome then goes to the membrane, and the endosome will fuse with the membrane. And this is where we ended up last time. And then out on the surface of the cell, there's this protein called MHC2 protein. And the MHC2 protein has attached to it, remember, this antigen fragment. This macrophage is now a professional antigen presenting. And this macrophage now will leave the site of inflammation, carrying this bit of some foreign protein, carry it off into one of the germinal regions. Could be in the tonsils, it could be in the it could be the tonsils, it could be in the Peyer's patches, it could be in the spleen, it could even be in the lymph nodes, any of those. It's going to go off into those germinal regions and present this antigen fragment, saying, anybody seen anything like this? And that is part of the immune response. So this is a professional antigen presenting cell. There are only 3 of them. Macrophages, dendritic cells, and naive B lymphocytes. And we'll come to naive B lymphocytes here in a minute. But this is where we left off. And what we want to do today is talk about how these professional antigen presenting cells will interact with T helper cells. And of course, the T helper cells are part of the branch of the immunity called cellular immunity. Let's make a table here about adaptive immunity. And we could talk about the idea of adaptive immunity coming as 2 flavors, if you will. One is going to be the cellular branch, and the other is going to be the humoral branch. The cell type responsible for the humoral branch are the B lymphocytes. Mature B lymphocytes are called plasma B cells, or B lymphocytes. These plasma B lymphocytes secrete antibodies. They secrete antibodies as glycoproteins. And the secreted antibodies bind to foreign proteins called antigens. To generate something known as an antigen antibody. complex. Largely what's happening with these antibodies is they're binding to the antigen and inactivating it. For example, the way antibodies are working for COVID is that they'll bind to the spike protein we talked about the other day. And then by binding to the spike protein, the antibodies will prevent the spike from binding to cells, prevent the viruses from getting into cells. But they have to form this antigen-antibody complex. The other branch of the immune system is the cellular branch. And in the cellular branch, there are T lymphocytes. T lymphocytes come as two forms. They come as T helper cells. These T helper cells are also known as CD4 cells. And they also come in the other variety of T killer cells. And these T killer cells all have another name called CD8 cells. So whether it's a T helper cell or it's a T killer cell, regardless, those cells had to pass through the thymus. They're T cells. So they've been selected for potency and specificity in the thymus. And if they've been correctly selected, then they go out into the world, as we talked about the other day. Now, these T helper cells and the T killer cells, what they in fact have in common is they have a transmembrane protein called a T cell receptor. The T cell receptor recognizes antigen fragments. Presented on other cells. We'll see that these T helper cells recognize antigen fragments. On professional antigen presenting. Cells. Whereas the T killer cells recognize antigen fragments presented on all nucleated cells. For these CD8 cells to recognize antigen on a foreign cell, the antigen fragment is presented on MHC1. Over here, remember these are professional antigen presenting cells, so these are now going to be on MHC2. So that's the general outline of where we're headed. Before we get there, we need to talk a little bit about antibody structure. So come up here, and we're going to talk about antibodies released by B lymphocytes. So these are going to be the glycoproteins that have been secreted from plasma B cells. We need to spend a little bit of time talking about this because these are the proteins that give the immune system its specificity. So that once you were immunized against any particular disease, but let's say it's COVID. Now, your immune cells are generating these antibodies. They're secreting these antibodies. And these antibodies all have a lot in common. So we're going to talk about here is antibody structure. When we look at one of these antibodies, we're struck by the idea that they're always shaped like a Y. And they have these two identical proteins called heavy chains. And then out here, on the short arm of the Y, there are two other identical proteins. These are again two identical proteins called light chains. So every antibody contains two heavy chains and two light chains. The two heavy chains are identical to one another, and the two light chains are identical to one another. Now, as a detail, these heavy chains, again, that are identical to one another, are held together by disulfide bridges. And the light chains are held to the heavy chains by disulfide bridges. Now, there's some more anatomy we need to delve into here. For each of the heavy chains, there's this long segment called the HC region. HC stands for heavy constant region. Now, we're going to talk about things called IgGs. It turns out there are IgAs, IgGs, IgMs, IgEs. We're going to focus on IgGs. If we're talking about all IgGs, they have this constant heavy region. They'll be the same for all IgGs. Then up here, there's going to be a region for the light chain. This is called the constant light region chain. So this constant light chain, C sub L, every antibody we look at, every IgG we look at is going to have the same constant light region. Now, where we begin to see some differences is out here on the short arm of the Y. And the short arm of the Y that I have shown here with this dotted line in orange, this is the antigen binding region. This is the region of the protein that binds the antigen. Now, out here at the short arm, there's the variable heavy region. And similarly, there's going to be on the light chain, the variable light region. The variable heavy will be the same on both arms. The variable light will be the same on both arms. But it's the 3D shape of the variable light and heavy chains that provides antibody specificity. So there's a particular three-dimensional shape out here that will fit precisely with an antigen. That's where the specificity comes from. And if this particular antibody, if we're talking about an antibody that we've generated to the spike protein of the COVID virus, then it's going to go out and cover up the spike protein of the COVID virus, preferably to the receptor binding domain. And it's going to block that receptor binding domain so that the spike protein on the COVID virus can no longer bind to our cells. That's how these antibodies are working. But to function in that way, we need to have a proper lock and key fit between the variable heavy region and the variable light region, forming a three-dimensional shape that will bind to that particular antigen. So any questions up to there? Yes?
Where's the variable light region?
The variable light region is out here on the end of the short arm of the Y. So this is going to be the variable light region in here. And this is going to be the variable heavy region in there. And we draw these in 2 dimensions, but they're a proper 3-dimensional protein because it's an amino acid sequence and it's folded into 3-dimensional shape. The 3-dimensional shape out here will be the same as the one over here because we have the same variable heavy region, variable light region, forming that portion of the molecule that can bind to the antigen. Okay? Anything else? I'll show you a couple of pictures then. This first image is one that could have been presented in about 1910. And what it's showing is something fairly simple, but it's an important or essential idea of immunology. So what we're seeing on the axis is time in days. What we're seeing in the ordinate is concentration of antibody in the plasma. Now remember, these are plasma proteins. They're out in the plasma. And so we're looking at the concentration of antibody. We see that with this arrow, this is the first exposure to antigen. So that could be a vaccine. It could be the organism gets sick with a disease. The antigen is presented at this point, but you see there's some delay. It's, a week or so before we get a peak in the amount of antibody in the plasma. So it takes a while to generate that response. That's why after you've had a vaccine, they tell you you're really not protected for a couple of weeks because it takes that amount of time to gin up your immune system to generate those antibodies. And we'll see how that gets done molecularly here in a little bit. But this is said to be the primary immune response. At this point, we get exposure. At this point, we then get proliferation. We begin to get proliferation of naive B lymphocytes. And these produce antibodies. They're secreting antibodies. It reaches a peak. We get over the disease. It comes back down again. But hopefully, we continue to have a resting level of the antibody. But what they're trying to show here is there's a second exposure. They're showing the second exposure about a month later, but it could be, if things are working well, it could be weeks, months, or years later. But with that secondary exposure, the response is way, way faster. So that the secondary response becomes stronger and faster. Rather than taking a couple of weeks to mount, this will be mounted in 24 to 48 hours. It will reach peak, and then you'll keep antibodies around for quite some time. This is one of the reasons why we have booster shots. We do this so we can get a secondary immune response and increase the amount of antibody. But then what we want to do now is we want to think a little bit about what those antibodies are, and then we'll loop back and talk about where all this comes from. This is the concept of antigen-antibody binding. It's a really crude cartoon, but I like it because it gives you a sense of what antibodies are doing. And it gives you a sense of the differential in size between the antibodies and the antigen. This antigen here is meant to be a gigantic protein. The antibodies are only going to bind to a region on that protein of about 16 to 20 amino acids. That region of the protein will have a special three-dimensional shape. So if there is a proper lock and key three-dimensional fit between the short arm of the Y of the antibody and the antigen, the antibody will bind to the antigen. But since these are fairly short regions of amino acids in the protein, there may be an amino acid sequence here that binds to this antibody, antibody 3, another short region up here that binds to antibody 1, and another one that binds to antibody 2. And what they're trying to show, although it failed here, they're trying to show there's a particular three-dimensional shape, the short arm of the Y, which now can bind to the antigen. So that's the idea of the binding, and this is this is a diagrammatic representation of what I just put on the board. So in lavender here is the heavy chain. These two heavy chains are identical to one another. In blue and sort of a grayish color here, those are the light chains. I showed them on the inside, these are the outside. Neither is accurate because in reality, this is going to be a three-dimensional structure. So this is the heavy chain, this is the light chain. Now up here is the antigen binding domain. This is the region of the protein that will bind to the antigen. There's a three-dimensional fit between the three-dimensional shape of the variable light and variable heavy chain. These guys are all intertwined with one another. And as they intertwine with one another, they're making a particular three-dimensional shape. And if this three-dimensional shape is a perfect fit with the foreign protein, the antigen, it will bind to it. And you can bind two antigens to every antibody. We'll see in the next slide, that means we can cross-link things. So this, I think, is the easiest place to see that, where these Ys can cross-link cells. They're showing erythrocytes, but you could show this for bacteria. So you could cross-link the bacteria, clump them together, and they could be attacked by macrophages. More commonly, we find that the antibodies will bind to the antigen and cover over the active site. And that's what I mentioned just a moment ago when I said that for the SARS-CoV-2, put your phone away, I'd appreciate it, for the SARS-CoV-2 virus on the spike protein, antibodies will cover up that region of the spike protein that will bind to the ACE2 receptor. And so we can hide those portions of the ACE2 receptor by covering them up with the antibody and preventing those viruses then from entering our cells. So any questions up to there? So that's the idea of the shape of the antibody. Now, what I want to do next is I want to ask the question, where do antibodies come from? So we'll take this panel. And we'll label it as the origins of secreted antibodies. And this is a cell lineage story. This is a story about how B lymphocytes are generated, how they contain on them a particular antibody, and how then those individual naive B lymphocytes are selected to proliferate and make copies of themselves. In the first iteration of the story, we're just going to focus on the naive B lymphocyte. And then we'll talk about the naive B lymphocyte as being a professional antigen-presenting cell. And that will bring us to the interaction between the naive B lymphocyte and the T helper cell, the CD4 cell. So we'll take this panel and label it as origins. Origins of antibody secreting. B lymphocytes. The origins of the antibodies secreting B lymphocytes all start out with something called a naive B lymphocyte. Last week we talked about the naive T lymphocytes leaving the red bone marrow. Well, the story's going to be similar at the very beginning here, that the naive B lymphocyte migrates to germinal regions of lymphoid tissues. It might go to tonsils, it might go to the pancreas, tissues. It might go to a lymph node. It could go to the spleen. But it will go to those sites and take up residency. Well, if we look at one of these naive B lymphocytes, so this is going to be a naive B lymphocyte. The naive B lymphocyte has a transmembrane protein out here as an antibody. This is an antibody. As a transmembrane protein. The story here is going to be pretty similar to what we talked about for macrophages. And then we're going to see that these naive B lymphocytes can be professional antigen-presenting cells. And so this transmembrane protein version of this antibody will be sitting on its cell in a germinal region. And along will come a protein. This will be an antigen. Now, if and only if there's a good three-dimensional shape between the antigen and the antibody, that starts the process of endocytosis, much like what we talked about with macrophages. So this cell starts to endocytose. It's hauling in the antibody with the antigen. Just as we saw before, it's going to package these antibody-antigen complexes in a vesicle. And these vesicles then will be guided, trafficked, to another membrane-bound compartment called a lysosome. Just as we saw with the macrophage, when this vesicle fuses, it's going to deliver the antigen-antibody complex to the lysosome. This antigen is going to come off. And in the lysosome, it's going to be digested into its component parts. And I'm going to label this as an antigen fragment. In the next compartment, this vesicle will fuse with the next compartment, which is called an endosome. And in the endosome, there's going to be this transmembrane protein, put it in orange, this transmembrane protein called MHC2. So this is MHC2, major histocompatibility class 2 protein, same as we saw on the macrophages, because these are professional antigen-presenting cells. So this MHC2 protein, just as we saw for the macrophage, is now going to bind to the antigen fragment. Then a vesicle buds off from the endosome. The vesicle contains within it the MHC2 protein with the antigen fragment. When this vesicle fuses with the plasma membrane by exocytosis, we now have this transmembrane protein here called MHC2, but the MHC2 is now displaying the antigen fragment. So we now have MHC2 displaying the antigen fragment. So all of that is reminiscent of the story we told about macrophages. But now we're going to introduce another cell. We're going to introduce a cell that's part of the T cell lineage. And what we're going to see, I may as well go to the upper panel. Take this panel, and in this panel, we're going to label this as the role of the T helper cell in naive B cell proliferation. It's a mouthful. We can say here the T helper cell facilitates naive B cell proliferation. So we've generated that cell above. We have this naive B lymphocyte. And out on its surface, it has this MHC2 protein, labeled as MHC2. It's a professional antigen presenting cell. On the surface now is this antigen fragment. Let me not put that there. So this is going to be the antigen fragment. Now the next character in this play is going to be this cell here. Let me put it in a different color. This cell is going to be the T helper cell, or CD4 cell. So this cell is going to be the so-called CD4 cell, also known as a T helper cell. It has a nucleus. One of its major transmembrane proteins is this protein that comes out here. It's called CD4. The function of the CD4 is to bind to the MHC2 protein. That's its only job. It's to bind the MHC2 protein and then hold these two cells together, moor them together. This is like a mooring line. These two cells now are going to be held together by the CD4 protein. Then there are two other proteins that become important here. This protein I'll show at this point. This is said to be the alpha subunit. This is the alpha subunit of the T cell receptor. Remember we said that the T cell receptors are transmembrane proteins? Well, that's the alpha subunit. Then there's a second subunit that accompanies it. This is the beta subunit. And together, the alpha and the beta subunits, these together, the alpha subunit and the beta subunit, form the T cell receptor. And the T cell receptor has a 3D shape that can bind to the antigen fragment. Bind to the antigen fragment on the MHC2 protein. So normally there's not a fit. 99.9% of the time there's not a fit. But if this antigen fragment has a good three-dimensional shape with this T cell receptor, what that does is it mobilizes a whole host of vesicles here in the cytoplasm. These vesicles come close to the surface, and they release molecules called interleukins. Usually it's IL-4 and IL-8, but there are a whole host of these interleukins. Then on this naive B lymphocyte, remember this is the naive B lymphocyte. There are transmembrane proteins here as well, and these now are interleukin receptors. The interleukins that have been released by the T helper cell bind to this interleukin receptor, and the receptor interaction, the receptor ligand interaction with the interleukins, this leads to cell division of the naive B cell. Each of the progeny, so this initial cell divides and gives rise to 2. This cell divides, gives rise to 2, and so forth. So this is the naive B cell. And these are the progeny. And after a few cell divisions, we go from having a transmembrane protein here as an antibody to eventually we'll see cells here where the vesicles contain, let me do it this way, vesicles contain secreted antibody. A better way of saying that is that the antibodies destined for secretion are loaded into vesicles. And when the vesicle fuses with the plasma membrane, these antibodies are released. So long and the short of that is, we need a T cell receptor to recognize an antigen fragment on an MHC2 protein. If there's a fit between the T cell receptor and the antigen fragment, then that causes the T cell to release growth factors, interleukins. The interleukins then tell this cell to start dividing, make copies of yourself. And every time you make a copy of yourself, make an identical copy of that antibody that we started out with. So in this way, we get thousands and thousands of copies of antibodies in each one of these cells, and each cell contains the same antibody. And eventually, we get these antibodies being secreted from the B lymphocytes. I have 3 minutes to finish the story. The final part of the story here is going to be T killer cells, and let me go through this quickly. T killers, also known as CD8 cells. These cells have a transmembrane protein on them called CD8. They also contain transmembrane proteins here, an alpha and a beta subunit. These are taken together are the T cell receptor. Now remember, the CD8 is going to recognize something called MHC1. So over here is going to be a garden variety nucleated cell. On its surface will be a transmembrane protein called MHC1. And the MHC1, just like the MHC2, the MHC1 is going to be displaying an antigen fragment. Just as we saw before, if there's a good fit between the T cell receptor and the antigen fragment, we mobilize vesicles. But now these vesicles contain something called perforin. When the perforin is released by vesicle exocytosis, the perforin intercalates into the perforin pokes holes in the target cell. Now, can't leave you with that, so give me just a minute. If you look at this the way I've explained it, this would kill all your cells. But that's not what's happening. What's happening is 99.999% put a self-protein out here. And there's not a good fit between the self-protein and the T cell receptor, so nothing happens. But now and again, you have a virus-infected cell. And if that virus-infected cell is now taking cytoplasmic proteins, mounting them on the MHC1 protein, and displaying them on the surface. So if this MHC1 is now displaying an antigen fragment of a virus protein, and that's recognized by this T cell receptor, then the recognition of the antigen fragment as a foreign protein, a viral fragment, then that tells the cell to release perforins and kill the virus-infected cell. But almost all the time, it's really rare that you're going to find a virus-infected cell you're going to kill with a T killer cell. But now and again, that happens. Because if you have a cell infected with the COVID virus and it's expressing COVID virus, fragments of those COVID viruses will be expressed on MHC1, and your T killer cells will recognize that and kill those virus-infected cells. And with that, I'll let you go. Thanks for the patience. Yeah.
This right here, did you say that's a transmembrane protein?
Yes, CD8 is a transmembrane protein. CD4 and C8 are both transmembrane proteins.
Okay.
And
so
like CD4, CD8 attaches not to the antigen binding site, it attaches to another site on MHC1 and moors the two together. So what I meant to do